All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, annyeonghaseyo, and hello all. And welcome and thank you, SynBYSS people, for joining us the 106th seminar, and I call it a UIUC Day. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Ting Lu. He is a professor of bioengineering at University of Illinois Urbana-Champaign, or simply UIUC. He is an excellent scientist and engineer with high-impact journal publication for many, many years. However, to me, he is a very kind person. When I had been suffering from many years of literally dying, physically and mentally, due to discrimination and harassment, he genuinely showed his sympathy and bought me even lunch during an ONR grantee meeting. We do not need billions of dollars or heroic actions to make this world better. A simple act of kindness or just giving a small thing such as your time or kind words can make big impacts on others' life and the world. Ting, thanks so much for giving me this amazing thing and contributing to society by your excellent science and research, and the virtual podium is all yours now. Thank you.
Thank you so much, Tae-so. Let me share my screen. Can you see my screen?
Yeah, wonderful.
All right. Well, thanks, Tae-so, for the nice introduction. And thank you so much for organizing these amazing seminar series, which have benefited all of us in the field of synthetic biology as well as system biology. It is my great pleasure to share with you our work on the analysis, design, and engineering of microbial communities. Microbes such as E. coli and P. aeruginosa are primarily single-cell organisms. However, in nature, they often form complex population and community structures. For example, on agar plate, E. coli can grow into concentric ring of spots. P. aeruginosa can develop finger-like patterns, and P. vortex can form spiral structures. In natural complex setting, microbes further grow into multi-species biofilms with a remarkable degree of complexity in space and time. So the overarching goal of my research is to dissect the assembling process and to control and eventually engineer ecosystem development. To achieve the goal, we employ a hybrid approach that combines mathematical modeling with experimental synthetic biology. Experiment can help to verify model abstraction and inspire new viewpoint. Meanwhile, modeling can help to analyze experimental results, generate a new hypothesis, and identify new experimental findings. The iteration between experiments and modeling can facilitate the understanding and application of microbial ecosystems. We are interested in synthetic microbial consortia because they can be used for different purposes. Synthetic consortia can be utilized as well-defined experimental model systems for us to uncover the basic rules of microbial assembly and functioning. In fact, compared to complex native microbiome, these like designing consortia contain a reduced degree of complexity and are highly controllable and amendable for quantitative measurement. Of course, synthetic consortia can also be used to create novel functions for practical uses, such as chemical synthesis, catalysis, environmental remediation, and others. In this short talk, I will give an example to illustrate the use of synthetic consortia for discovering the role of microbial organizations. Microbes are highly interactive in nature. They can communicate with each other at distinct spatial scales. Some can occur over remote distances through the release of diffusible small molecules, such as quorum-sensing molecules and membrane vesicles, while others may interact with each other in close proximity through direct physical contact, such as C signaling in Myxococcus. Apparently, both types of interaction are present in nature, which lead to the following question. That is, how does the spatial variation of interaction drive the development of microbial communities? To answer this question, we engineer a pair of synthetic two-string consortia. As shown on the left, one ecosystem involves a green string which produces quorum-sensing molecules to trigger the lysis of the red string to confer a long range of interactions. The other ecosystem on the right-hand side is composed of a green string that directly injects toxin to the red string through physical contact. So in both cases, green string kills the red. However, they're different in space. One through long-range diffusion and the other through contact-dependent interactions. So using fluorescent microscopy, we validate the two engineered ecosystems. As shown here, we found that in contact-dependent inhibition case, the green string is able to kill the red when they're in direct contact. However, when they're apart, both can grow over time, as shown on the left columns. By contrast, in the long-range diffusible case here, the green string suppresses the growth of the red, with or without direct contact, as illustrated in the middle columns. For comparison, when the interactions are absent, the green and red string are both able to proliferate, even though when they are in direct contact. So with the validation of the ecosystem at single-cell level, we perform the colony expansion experiments on agar plate by adding a droplet of mixed strain onto agar plate and watching the growth of the colony over time. The results show that without inhibition, the green and red strain occupy roughly equally at the growing front of the colony, as shown on the left two panels. However, in the presence of contact-dependent inhibition, we found that the green toxin producer is more abundant, and after 48 hours, it dominates the entire growing front. By contrast, when there's a long-range diffusible interaction, we found that the toxin producer is able to establish the dominance within 12 months, as illustrated on the bottom right corner of the slide. So when we compare the final image of the colony with different interactions, we found that there is a reciprocal relationship between spatial interaction scale and the time required for the toxin producer to establish a dominance. That is, a longer spatial interference scale caused a short time for the sensitive to extinct. To rule out the possibility that the space-to-time translation for competition was caused by different growth rate of strain, carry out a different fluorescent reporter, we actually swap the color of the strain and develop a system where red strain kills green. As shown on the bottom row here, the result demonstrates that this space-to-time translation is indeed caused by a spatial scale interference, not by different reporters. This synthetic consortia we showed above involve one direction interaction where green kills the red. However, in nature, microbes often interact with each other in both directions. So therefore, we create a new ecosystem involving mutual inhibitions between two strains. As shown on the left, here now the green strain suppresses red through short-range inhibition, while the red strain inhibits the green via long-range inhibitions. We develop a mathematical model, and using the model, we predict that in the presence of bidirectional interactions, the community structure diverges into distinct monocolony structures and different initial conditions. Indeed, our experiments on the right-hand side confirm that it was the case. We observed a monoculture green or monoculture red colony during such bidirectional interactions. And such kind of colonies for morphology is given by the underlying interactions motif and also are subject to total cell density and the relative ratios. Again, so we swap the color of the two strains and to rule out the possibility of growth difference because of fluorescent reporters. And we found that the result remained the same, which demonstrates that the divergence of colony structure was not caused by different growth rate. Additionally, we noticed that at the boundary of initial colony conditions, we observed the colony to have incomplete or sometimes weird morphology. This was because in these conditions, the two inhibitions are comparable and the colony structure are mainly driven by stochasticity or noise in gene expression and the growth dynamics. That is why we see different colony pattern when we repeat experiments. It illustrates on the right-hand side. So together, we show that engineered consortia can be used to advance our basic understanding of microbial organization in space and time. So in addition to being used as a model system as basic understanding, synthetic consortia can be utilized as a powerful tools for different biotechnological and medical applications. For example, recently, we engineered a consortia of Pseudomonas putida to efficiently upcycle PET, a common plastic used in water bottle, into valuable chemicals through design of division of labor among microbial strains. In another case, we engineered a community of lactobacillus and Saccharomyces cerevisiae to produce a 2-butanol, which is the secondary alcohol that used as a precursor for the industrial solvent butanol. I hope that through this very short talk, I could show you that synthetic consortia are powerful tools. They can use as a well-defined model system to understand the basic rule of microbial ecology. Meanwhile, they provide as powerful tools to enable biotechnological and medical applications. And I would like to thank all my students, collaborators, and with the funding agencies. And thank you all for your attention.
So thank you, Ting. I mean, this is absolutely wonderful talk. You show amazing consortium, of bacteria could do many things, including plastic, even degradation or upcycling. And also you show that using collaboration, that is a human consortium, I guess. So thank you so much. I mean, this is absolutely great talk. Thank you. All right, so now the main speaker of today with a longer introduction, Dr. Carlotta Ronda grew up in Italy, where she graduated with a degree in molecular biology from Padua University. She pursued her PhD at the Technical University of Denmark, DTU, in the Novo Nordisk Foundation Center for Biosustainability, where she was among the earlier developers of CRISPR technologies for multiple organisms. After completing her PhD in systems biology, she moved to Columbia University in New York City as a Simons Junior Fellow in Harris Wang's lab, where she developed new gene editing tools for modifying microbial communities in vivo and microbiome-based therapeutics. She develops novel models and molecular systems to investigate the fundamental basis on how microbial communities and their hosts communicate and shape their ecosystem. She also applies multi-scale programming of microbial communities to develop novel therapeutics. She also spearheads the development of an anaerobic co-culturing system of human gut organoids with microbial communities and new genomics technologies applied to stem cell engineering and human organoid. She's now at UC Berkeley as an independent researcher, and I will meet her this week at the Microbiome conference that I have been organizing with other great chairs, and she is actually one of the invited speakers. That means she's a really true rising star. So Carlotta, thank you so much for your time today. Please take it away. It's all yours now.
Thank you so much, Tak. Silk, is an honor to be here. Thank you for inviting me. I feel excited to discuss with the community the work I've been doing in the brilliant lab of Harris Wang, and then how I will continue with my future in the scientific endeavor at Berkeley as a principal investigator at the Innovative Genomics Institute and then adjunct professor at the UC Berkeley. So I want to discuss today the power of synthetic biology to be able to edit and help us to map the human microbiome interactome, which I'm personally very interested in to use synthetic biology to apply to discover new mechanisms and to apply it as a novel therapeutics. And so let's start from the bacteria. Let's start from the stars. So the microbial communities actually represent half of the biomass on Earth. They are everywhere. We Professor Lu has just discussed about the incredible work he has done in the biofactories and environmental component, but they are everywhere. They're used as a fermenters in pharmaceutical to produce pharmaceutical. They are involved in our health. We are not exempt from the community to be part of our daily life and to be actually part of our own physiology and our own life, in terms of they are able to dictate or to influence the chemistry along the all axis of the body. We have microbiome everywhere. We have microbiome in the gut, is the most notable one, but we do have a microbiome on the skin, in the surface of the head and the eye. And so the microbiome is something that has been neglected a little bit in the past as it just helps us to digest. But now in the past 10 years, we have been rediscovered the microbiome has the new genome, the pan genome that we were missing. And so now we need to have a holistic approach. When we look at the physiology of human physiology, we can just look at organs and tissue by themselves. We need to consider the community that co-evolve with us, and they are our holistic genome. And so when but this community in our body, they are in dysbiosis, so they are not in homeostasis with our physiology. They are in fact able to trigger numerous disease. And so my lab, or I've been very interested to understanding this community and understand how their metabolism, what are the mechanisms they basically allow them to have this strong effect in our daily life and our physiology. And the way we've been looking at community, or the way that we currently have been studying community, is through these like incredible, breakthrough ability to sequence them. So we do next generation sequencing, and we look at the compositions and the phylogenesis, or like the phyla of this community, like what are the players in the community. But this just gives us like a snapshot in time of what actually is happening. Communities are dynamic entities that they constantly are fluctuating in different states. And so just looking at this snapshot in time of that specific sequence and that specific compositions in terms of, in terms of phylogeny, it doesn't really give us the resolution at which we can understand their implications, their effect in our physiology. And so this is what I've basically driven my research in have me embrace this system and synthetic biology approach, is because I really want to understand what are the molecular mechanisms that regulates these like interactions with this, our body with interacting with this community. And so and how can we increase the resolution at which we can look at this community? Can we use synthetic biology to look at, to put a microscope on this community, no pun intended, and then, manipulate them to eventually being able to use them for our own good, but also to understand them. If we have a stable, steady state, we won't be able to understand the community unless we perturb it. Because if we look at it, the complexity of this community, it's such a multi-layer approach that people with my background in systems biology and synthetic biology are, the way we approach this problem of multi-layer of complexity is to being able to basically break down this system of nodes and complexity into small little single problem. So if we can manipulate in a more systematic way this complex network in a way that allow us to perturb them, and for example by inserting new functions, and then we are able to basically start to understand and how to deconvolute this network. Because if we are able to modify this node in this network, we will be able to eventually understand how they work, how the whole community responds to changes. And then if we infiltrate functions, we can eventually also introduce novel perturbations that can allow us a deeper understanding of this community, and also in terms of future ideas. In terms of therapeutics, this has like a great impact in terms of medical implications. And so, and then using, a synthetic biology approach, we can eventually also propagate the functions within the community, which create a ripple effect that will allow us to better understand and dissect every, the functions of at the gene level, at the phyla level, how this community interact with us and interact with one another. The players in this community interact with one another. So, the way that we have been looking at this community in the past, beside the sequencing, that gives us this very precise snapshot in time, we have been manipulated the community as being introducing these very sweeping changes, adding antibiotics or probiotics or fecal transplant, because we didn't have methods, or we didn't have genetic tools that allow us to proper manipulate these complex networks, this node of intricate interactions. And so just by introducing something like a antibiotic wipeout that only few species could survive, then we could be able to start to perturb this community and trying to understand their functions in a specific context, for example, disease. But this just doesn't give us the resolutions we want. And that's where I think synthetic biology plays a key role in to help us to really understand these specific problem. And so creating new tools for bacterial community is at the core of understanding the microbiome, to then eventually use this knowledge to create novel therapeutic and to better understand the human physiology. So we need to go towards a targeted microbiome editing, because by using targeted microbiome editing, we can specifically modify at specific nodes in this network that allow us to better deconvolute the complexity. And so during my my work at Columbia in Dr. Harris Wang lab, I have developed 2 genetic tools using synthetic biology with this purpose. The idea that if we are able to find ways to create efficient, specific tools for non-model organisms, and also then deliver them in a very efficient way directly in situ in their own native environment without disturbing or disrupting the milieu like you would do with antibiotic, for example, then we will be able to begin to understand these communities. So I developed, in collaborations with the Sam Sternberg lab, a tool that is based on CRISPR transposase that we'll call integrate, and then and I developed also a tool that allow us to directly in situ manipulate the microbiome directly in vivo in mammalian gut. So the system that I developed to directly engineer community in vivo is called MAGIC, that stands for metagenomic alterations by in situ conjugations. This system was able to infiltrate the native community of the human gut, the murine gut, sorry, and was able to engineer and modify directly all 4 major phyla in the gut. We were able to modify strains that they're not currently cultivable, and also some of them they were able to cultivate. We were able to isolate them. So the power of this system is that even though the bacteria, they are like in microbiomes that are not cultivable, they might be still genetically tractable. So using MAGIC, we can engineer directly bacteria in situ without the need of isolations, which is a huge burden or a huge hurdle actually for any person that works in wild or non-model communities. In native communities. And I, as a follow-up, I wanted to increase the edit specificity, because the system, a MAGIC system, is a system that is based on bacterial, it's based on conjugation. So it's a bacterial system that it's a repurposing of a bacterial system that is able to mobilize DNA from one species to another. But it was lacking a little bit of specificity. So in order to increase the specificity, I went to what we know are the most specific genetic tools known to mankind at this moment in history, which is CRISPR systems. But we know that they are immune system for bacteria. So they are extremely toxic. Cas9 is extremely toxic because they induce double-strand break. So I developed a system that uses a different type of CRISPR, which are CRISPR transposon, that they don't introduce double-strand breaks. And so so when we introduce the modifications, we are not going to kill the bacteria, they're going to survive. And also they're more portable to different species, because they're not dependent on the repair mechanisms like, for example, Cas9 and other systems are, and other CRISPR system are. So this system, we call it integrate, that stands for an insertion of transposable elements by guided RNA assisted targeting. It's a very long name, so we came up with integrate for short. And this system is, just in short, it's a type 1F CRISPR transposase that we repurpose from Vibrio cholera, and then we tested it in E. coli, and we were able to sorry, we were able to insert up to 10 kb in the genome of E. coli using this system. So this system also has the incredible ability to insert large stretch of DNA sequences. So if we're thinking about engineering community, if we wanted to infiltrate functions, this is a fantastic system to introduce novel functions when you have, for example, operon or large genes that you want to introduce. So these offer this amazing possibility of, cargo size to be introduced, or functions of the cargo size of the functions you wanted to introduce. And the beauty of this system is that it's also extremely precise. It's chirurgically precise. So when we tested in E. coli, we had like 99.8% efficiency in integrations at the target site, because it's a CRISPR system, is a marriage between a CRISPR with the specificity of CRISPR and a transposase, and so the ability to mobilize large fragment of DNA and into the genome. So the combination of the two has created this incredible tool for synthetic biologists to being able to being extremely precise and introduce large size of DNA. Additionally, this system is also portable to other species, which means that it doesn't really rely too much on host factors, which means that if we wanted to work to engineer microbes in community, this is a perfect system because it's precise and is promiscuous. It can work is promiscuous, so it works in different species because we tested in the clinically relevant strains like Klebsiella oxydoca, and in more industrial and in more industrial and in more industrial strains like Pseudomonas putida that is used in biotech and for productions of compounds. And so when we married this system between MAGIC, which is the ability of efficiently deliver in vivo and engineer species in vivo, with the high programmability and specificity of a CRISPR system that is able to mobilize a large, stretch of DNA, then we have a very, very powerful system that we called it Magicast that allow us to now dive into the community and then introduce functions and knock out genes directly into the community. So we went and we tried first in vitro and to target a complex community. We took communities from different cohort of mice. And then we decided to target a specific gene, and that was we're targeting a Klebsiella the specific gene that was in Klebsiella oxydoca a genome. And then when we went in sequencing, we saw a remarkable efficiency in the ability to edit this community. And it was a very specific editing because nothing was edited that it was in Klebsiella oxydoca at the specific target site of LAC-Z that we actually picked for this gene. And so this is an exciting tool that gives us a targeted microbiome engineering directly in situ into the active native community. And so now, if you think about it, we can start to build a system, and here synthetic biology and the synthetic biology community can, go crazy with like the type of modularity that and the type of additions that we can put into this system, because it's been built in a modular way. So because MAGIC is a modular library of vectors with CRISPR transposase that we have created like a modular vector as well. And so we can just start to become to start to dive in and begin to understand the functions of different species in the communities that we couldn't modify directly in vivo in a specific way before. So in our case, we went and decided to customize our Magicast system for different type of commensals. So we had a gigantic, we have one of the most updated human isolates database, sorry, in Dr. Wang lab, that like there was a huge culturomics effort, and we were able to cultivate a large number of species. And so we customized these Magicast systems for different type of species. And I want to bring you here an example of Bacteroides, which is one of the most wanted commensal, with Clostridia is one of the key ones. They are the key species that they are some of the most studies for the interesting functions that they are playing in our physiology. And so we have customized this system for these Bacteroides, and we decided to test them in wild isolate, from humans isolate, that we had them from this database. And then when we tested in different species, we were astonished to see that it works remarkably well at very, very high efficiency. But the incredible component is extremely efficient into target only this specific gene that you designed the guide RNA for. Indeed, if you can see on the right, those are I don't know if you can see for on the right side, there are basically, I can't find the pointer right now, but on the right side, you can see there are all the reads that they map after like a whole genome sequencing, like next-gen sequencing, all the reads that they map to the genome of these wild human bacterial isolates. And you can see the peak where all the reads where our targets is inserted, and is extremely precise, over like 90% integrations efficiency. This work has been done with in collaborations with the brilliant postdoc as well, Diego Rivera Gossinger, and then so when we look at it into this efficiency, we were wondering, can we just move these like incredible efficiency that we were able to detect in vitro, can these be something that works in vivo? And so we moved our system in vivo. And so we decided to use Magicast, where we use magic, which is the ability to infiltrate and deliver DNA and engineer specifically in vivo community, with the targeted precision of integrate. And so Magicast, we targeted a specific gene, and we decided to knock out that gene that we are interested in to study the functions in a wildlife community in mice. And we develop a specific target-seq pipeline that allow us to basically detect a very low level of editing efficiency in full raw community, like our insertion site, and if it's actually precisely targeted inserted. And so we were very excited to see that when we target a specific B-Theta or Mullobacteriaceae, like it's a specific strains in mice, directly in vivo, when we deliver Magicast to mice in mice gut, after just 6 hours post-gavage, we were able to see that we were indeed editing the community, and our edits were extremely on target. So above 98% of our reads mapped to the target site that we were indeed interested in knocking out. So this system has brought us has excited us to the idea of also like applying it to different type of different type of scenario, different type of field, different type of applications better. And so this is a fantastic example of how can we use this system to directly manipulate native community. This community have not seen any antibiotic, have not seen anything. They're just native raw community. We were able to achieve targeted microbiome editing. And so then we wonder, can we use this system also for therapeutics? Can we find the therapeutic applications? And so we were interested in these pathogens that is enterohemorrhagic E. coli, and I've done this work with two talented grad students, and Tyler and Purdue and Lagerschwanz, that like we have been thinking, okay, so can we develop a system that can we use Magicast for translational applications? Can we target a specific virulence gene into these enterohemorrhagic E. coli that we know it can still cause death around the world, and so it's a very toxic strain that contains what is called Shiga toxin. Is a very strong virulence gene that when it's activated, you have you basically have the strongest phenotype. So we wonder the stronger clinical phenotype. So we wonder if we could use Magicast to swap this toxic virulent gene and then make this strain basically non-virulent, and then insert, because Magicast can insert payload. That's the beauty of it. You just don't cut DNA like Cas9 and kill the cell. You just can insert and knock in, so knock out and knock in at the same time, a gene that recruits the immune system, and it becomes basically a self-bomb, like a ticking bomb internally into these toxic strains. So not only it becomes a non-virulent, and so you supposedly won't have any, symptoms of pathophysiological effect, but you also will recruit your own immune system to kill it. And so we tested in vivo in mice. And so after just 24 hours, 2 days roughly, we reach almost 100% of swapping the toxic gene to a self-killing gene into these strains. And so we for the first time, to our knowledge, demonstrate in vivo real gene drive out a toxic trait in these strains and making them non-virulent within in the populations within less than 2 days. And so when we look at the mice that they had the knockout strain versus the wild type, this preclinical model in mice is extremely potent, or like it's very extreme in the sense that the mice, if they don't get this virulence gene knockout, they die, they succumb within 5 days. But when you knock out the gene, then the mice survive. So that means that this gene is the major driver of the toxicity of these strains. And at the same time, you also recall the immune system. So when we look and we zoom in more specifically into the efficiency over time of the system, so on the left, you can see the conjugation's efficiency reach eventually 100% in vivo, and we have a clear reductions of the toxin, like MG6055 is our vehicle strain, and then the STXB XTX2B is the targeting. XTX2B is actually the gene name of the toxic gene. It's actually the strains that carries Magicast targeting the XT2B toxin gene, the Shiga toxin gene. And so we can see that over time we have a striking reductions in even though the data are very noisy, we have a huge reductions in Shiga toxin productions. So this system basically allow us to have the immune system to react to the pathogens that it has become inactive, has become kind of like an attenuated strains. And then when we reinfect mice with the virulent strain. So the mice that they got they did have the knockout strains, and then they survive, and they did not get any pathological symptoms or pathophysiological phenotype. Then they were reinfected with the virulent strain that had the toxic gene again, and this time there was not Magicast, but they were infected twice. They actually did develop an immunoresponse. So it seems that this system not only is able to knock out the genes that cause the strong pathogenic phenotype and make a non-virulent these strains, but the recruiting the immune system makes the immune system to remember that strain very strongly. And even after repeated reinfection of the pathogenic strain, the mice, they are immune to actually these specific toxic strain. So we have, these data show you like the potential of Magicast in translational application. And so I want to just like spend a short time at the end of the talk to trying to give you an overview of like what you can achieve using Magicast and what I believe we can achieve using Magicast. So if we use this system to perturb community, now we can, and we combine this system with omics technology. So we combine synthetic biology with systems biology. Then we are able to truly increase the resolutions at which we can understand this bidirectional signal between the host and the microbiome, and how these like commensal coevolutions with this community plays a crucial role in our daily life. And so using Magicast, we can insert different type of functions, and then we can look at how the community in different background, could be clinical disease, will react, or even just steady state, will react to that specific perturbations. And this is something that my lab will continue and pursue. And so in terms of therapeutic applications, when we think about Magicast, I believe that I wanted to see that we can basically achieve a personalized type of transkingdom gene therapy, because then at this time we can use Magicast, for example, to insert a gene and have like a protein biofactory directly in your gut that basically produce protein that your body doesn't produce anymore. Or we can insert therapeutics that have an effect locally in the gut, or they can express genes, because we can insert genes that have an effect in the brain through gut-brain axis, or for example, that have a overall systemic effect, maybe affecting the immune system. And so we can use Magicast, for example, to do metabolic pathway replacement. I'm thinking about like there has been this successful and incredible company, Synlogic, has done this incredible work with phenylketonuria, which is a metabolic disorder that like people, they're not able to degrade phenylalanine, and then they have a terrible quality of life. They have to have special diets, and it's lethal if you don't follow a specific diet. And so it's chronic, it's forever. And so they developed these probiotics that is able to basically replace metabolically the missing enzymes in human. And so why not using Magicast to directly insert it in and then have it forever, clinical, therapy that will stay in your body, in your own microbiome. And so it's your own personalized therapy. And so we can also, use Magicast as make like personalized therapy by just like, for example, retrieving the microbiome, use select the best performer with Magicast that have the best metabolic background to perform the functions that we're interested to insert in the patients, and then select them and then redeploy them back as a personalized transkingdom gene therapy. And so I want to try to put it into the community, the idea that I think the microbiome is going towards that directions. We can believe that the microbiome will become maybe the next type of gene therapy, where we're not editing our own genome, we're editing our pangenome or accessory genome in the case of the microbiome. And so with this, I actually would like to thank and I'm extremely grateful to my postdoctoral advisor, Harris Wang, who has gave me an incredible playground in New York City at Columbia to, achieve all of this work. And I'm like eternally grateful. And then the incredible people that I've been able to work with, like the entire, Magicast team. So Diego Rivera-Gelsinger, Tyler Perdue, Logan Schranz, and then the Sternberg Lab, who I have collaborated with Leo Fo to develop the integrate system. And then I want to close. I think the entire Wang Lab is being a fantastic experience. But I want to close with the idea that I'm now moving forward, and then Ronda Lab is starting, and there is not a better place than here in the community of synthetic biology to announce it, that is opening its door at UC Berkeley, the Innovative Genomic Institute, and we are going to, trying to dive in and map the human microbiome interactome, and we're going to use in vivo engineering, like the systems that I have presented today, and I'm going to introduce soon at one point a new co-culturing system using human gut organoid, where we can, looking at the real human host interactions with the communities, and all is developed so we can have anaerobic chambers integrated in this device. And then we're gonna develop, new type of therapeutics using synthetic biology, like programmable live therapeutics. And so with that, I really wanted to thank the TechSale for the invitations, the community for listening, and then this team, Professor Lu, that preceded me with his wonderful talk, and I'm open to any questions.
Wonderful talk. This is really great. So you achieve a lot, and then also I'm a big fan of the Harris Wang's,
He's fantastic.
The work. So it's fantastic. So I'll start with some questions. So you show amazing data, even after the infection, you see the clearance of this pathogen. That's a wonderful data. And I now see the huge potential of your approach or technology that, for many other applications, including not only pathogen elimination, also some other application. At the same time, one thing we need to address, as you probably know, my lab also developed some technology similar to what you did, but that's the strain level targeted, genome, microbiome engineering, tool development and so on. But at the end of the day, one thing we need to make sure is we don't want to have, those engineering tools potentially lead to generation of course, we're generating so-called GMO, and then we also need to ensure the biocontainment. At the same time, other concern would be your efficiency of your tool is amazing in terms of the horizontal gene transfer is not that much efficient, cellular level, but you showing the amazing efficiency in general. So I think, some challenge we need to overcome. And then what I want to hear from you is basically what kind of remaining challenges we should think about and then overcome to make this technology, fundamental technology, from the laboratory study tools to real translation, including you mentioned the PKU and so on. I just want to hear from you about those kind of aspects.
Those are fantastic questions. And then there are like impelling questions for the whole community. Like biocontainment is huge. Like if we are editing the microbiome, it's important to have biocontainment. And it's something that I feel that like the microbiome field is more approachable, at least for the FDA, to editing. If you think about right now, gene editing is a huge, there is a huge ethical debate if we can edit human, and that's a problem, because even if it's not germline, like the precisions in vivo of CRISPR, it's always like a problem. It's always like a challenge. And so by using the microbiome as kind of the platform where like you're missing an enzyme, you're missing a functions, and then you're not editing your own genome, but you're editing bacterial genome, it's more accepted, I think, in terms of both by the FDA and also in terms of like public, opinion. And I feel that and the biocontainment here plays a crucial role, because if we are editing this microbe to make people feel better and the FDA feel better, we have to have ways that we actually biocontain these specific organisms. And the beauty of having bacteria is that we can introduce genetic circuits. And we are still behind in the ability to edit in a very efficient way, no model organism. So yes, the efficiency are very good, but they are in a laboratory setting. They are with mice, and I'm the first person that is excited about the data that we have, but at the same time, I'm also very careful about the data that we are generating, because as you mentioned, you are in the laboratory setting. Is it different than if you wanted to move in humor and in vivo? But definitely continuing to work as a community to develop tools that allow us to work with no-model organisms and make them more efficient. This is an effort that the whole community should work on, and it should be a communal effort to actually being able to achieve that, so we can insert like a very precise and very tunable genetic circuits. I'm thinking about promoters. Even promoters, they are like, we don't have enough promoters. We're like, one of the major challenge that I have when we were designing these like customized vector is like, we don't have parts for most of no-model organism. This is a huge area of synthetic biology that we should tackle, have like array of like a study promoter, study regulatory systems that we can have precisions in the way we design our genetic circuits, and modularity that we can fine tune them. And then in terms of clinical implications, I think we are far from the clinic yet. It's in mice, and it's in a different setting, and then it's a human isolate. So we had to have a pre-treatment with someone with antibiotics to have it to become so virulent early on, because otherwise they would not stick into mice. So it's as a laboratory setting, it's a great data, it's a great initial it's very hopeful, but it's way not even close to the idea of getting into clinic. And I think the major hurdle for editing communities that I find that is really something that is challenging is the delivery in vivo is one of the most challenging one of the most challenging things in I would say in any area of research that has genome or genetic editing. Even if you look CRISPR, one of the major problems is actually delivering Cas9 to in vivo. And the same is for microbiome engineering. Delivery is, there are so many hurdles to get the DNA. So to get the DNA, to get the donor cell to getting into the recipient cells, the DNA in and stably in, and then express, because we know that there is residual methylation, CRISPR, immunity systems in commensal, that's like a barrier. So you have multiple barriers that you have crossed. And if we think about it, we don't have many systems in place. We have very few conjugative systems known.
Right. That's wonderful. So, you know, that's fascinating. So actually in our case, we use the liposome to deliver this DNA, the CRISPR system. But still, you know, I'm excited because, you know, UK, you know, for the first time approved CRISPR, you know, based technology for human, you know, application to, you know, cure some disease, genetic disease. But at the same time, as you mentioned, you know, if we manipulate microbiome, that might be safer in some sense, but probably we also need to have the careful investigation, the safety and whatever in the manipulating microbiome, because still we need to know a lot more because we still a little bit early stage in terms of this type of research and development. So fantastic. I mean, I hope you provide the solution in near future in all the problem we're thinking about right now, and this is an absolutely great one. So let me check with the question from the audience. This is already 6 minutes after 11, so
oh yeah, sorry, I was like,
it's okay.
I pitched in my lab since I just started, and this is in a fantastic community. So
Sure.
I thought it was a great audience to, recruit.
Yeah, sure. So
Thank you for inviting
let me close by saying the last thing. Okay, so amazing talks. That's this brilliant one. I really also excited by your talk. So thank you all for joining and staying today, even though many people busy with the final exam and so on. We'll meet again next week on December 14th, Thursday at the same time, the same Zoom link. We'll have the Professor David Savage at UC Berkeley and Professor Sung-Sun Lim at KAIST, another Harris Wang laboratory alum. Interesting coincidence or interconnection for the last seminar this year. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty face if you wish. And thanks. I will stop recording. Just give me 1 second.